Good morning, good afternoon, good evening, everybody. My name is Amaicha de Pino from the University of Buenos Aires, Argentina. On behalf of the Young IBRO committee, I want to welcome everybody to our second NextInNeuro webinar series. We want to let you know that this webinar will be recorded and available afterwards in the webpage of IBRO. You can ask your questions in the Q&A section in Zoom, and we will read them at the end of the talk. Those of you who need a certificate of this webinar, you will have a link to request it in the chat at the end of the seminar after a short survey. The mission of Young IBRO is to foster the interactions between IBRO alumni and help young neuroscientists to get together and discuss the neuroscience we want. We do so taking into account IBRO's core values, with a special focus on diversity, equity, and inclusion. If you don't know, we are hosting two types of grants: Young IBRO Connecting Awards once a year, and Young IBRO Maternity Parenthood Grant twice a year. You can check them up in our webpage. I now let Dr. Aparna Subratan from McGill University in Canada, also a member of Young IBRO, to introduce our speaker.
Hi, everyone. It is a really great pleasure to welcome Manuella Kaster to give this NextInNeuro talk, the second in our series. Dr. Kaster is currently an associate professor at the Federal University of Santa Catarina in Brazil. She is an affiliated member of the Brazilian Academy of Sciences and is also a young global leader from the World Economic Forum. To briefly describe her academic path, Dr. Kaster completed her PhD also at the Federal University of Santa Catarina under the supervision of Ana Lucia Rodriguez. She then went on to a short postdoc at Rodrigo Cunha's lab at the University of Coimbra, and then a second postdoc mentored by Kai Guzens at the McGovern Institute for Brain Research at MIT and at Mount Sinai. So Dr. Kaster's research investigates the immune system and stress with a focus on stress resilience and mood disorders. She studies the mechanisms underlying these phenomena all the way from the molecular to the behavioral level, with research both on animal models and at the clinical level. Dr. Kaster has published really fascinating research on topics that range from caffeine and its effect on adenosine receptors in the context of stress to how inflammatory cytokines can produce depression-like behavior. So we're really looking forward to her talk today on CD300F immune receptors in mood disorders. So we're very excited to welcome you to this, and we are looking forward to your talk. So please join me in welcoming Dr. Manuella Kaster.
Thank you very much, Aparna, for this kind introduction. So just let me share my screen. So good morning, good evening, everyone. My name is Manuela, as was already told. So I'm very, very happy to be here, to be part of the IBRO community. And I'm from Florianópolis, which is a small city in south of Brazil, and I work in the Federal University of Santa Catarina. And as Aparna told us, my research, I coordinate a small research group that focuses on two different lines. One is how cholinergic signaling actually modulates behavior responses and emotional behavior relevant to psychiatry disorders. And the other one is more focused on neuroimmune mechanisms, which is part of the results that I'm going to show you today. And before I start, I would like to thank the International Brain Research Organization, because actually this whole results started with IBRO grant. So a PhD student at my lab, Fernanda Kaufman, was awarded with two IBRO grants, a travel award and a short stay grant to visit another lab in Latin America. And she went to the lab coordinated by Dr. Hugo Pelufo in the Pasteur Institute from Montevideo. And that's how we start. are actually the immunologists. We are more focused on the neuroscience. And that was the start of really, really important and really good collaboration for us. So thanks to IBRO, we are able to start this work. So I want to talk a little bit today about these receptors, CD300F receptors. Most of you are probably not familiar with them. They are not well studied. And these receptors are highly expressed in microglial cells. And we are investigating their ability to control not only microglial metabolism, but also mood responses relevant to psychiatric disorders. So I'm going to talk a little bit about psychiatric disorders, actually major depressive disorder and depression. That is our major focus on the lab. And I think that this is a really necessary first slide in any talk about depression. We always ask ourselves, why do we worry about this condition? Why do we worry about psychiatric disorders? And it's really, really easy to see why we do need to worry about that when you look at the prevalence of this condition. So according to the latest World Health Organization data, depression affects over 300 million people worldwide. And the prevalence can range, the lifetime prevalence can range from 16 to 32%, depending on the type of study, but the prevalence is really high. And it is a highly debilitating condition. It's already the first cause of disability, and it starts relatively early in young adults mostly, and it can last a long time. And in addition, it is a life-threatening condition, and nearly 30% of all patients with depression have suicidal thoughts or suicidal ideation. So it's a huge public health concern. And I believe that we have to worry even more about mental disorders after and during and after the COVID-19 pandemic. And this is the studies evaluating the impact of the COVID-19 pandemic on mental health are still emerging, but it's already safe to say, and here there's some data from UK and United States, showing that symptoms of depression and symptoms of anxiety are actually increased a lot in the population during this period. So it's something that we have to be concerned. And when we look at this scenario, one thing that it's interesting is that psychiatry is still a little bit behind other areas of medicine in terms of a precision approach to classify and to diagnose the patients. And today, the diagnosis of major depressive disorders is based entirely on the symptoms, on clinical phenomenology, and the patient has to experience at least one episode of depression for 2 weeks. And these are the symptoms that are evaluated. The symptoms were in accordance with the Diagnostic and Statistic Manual of Mental Disorders. So the patient has to experience at least one of these two symptoms, that is depressed mood and anhedonia, or loss of interest in or pleasure, and 4 of the following conditions that include alterations in cognition, alterations in somatic symptoms, and others. And when you look at this picture, it's safe to say that we can have patients experience different combinations of symptoms. And this is true. We have here this study that did the math, and there are 227 possible combinations that still meet the criteria for depression, for major depression. And we do know that there are some combinations that are more common than others, but it is still, when you try to understand the biological basis of this condition, it's a nightmare, because we have many, many different systems and many different types of behaviors involved. And in addition to all that, depression has high comorbidity rates, not only with other psychiatric conditions, but also with clinical conditions, with metabolic conditions, with heart disease, and others. So we need to study more the biological basis, and we need to understand this heterogeneity to have better ways to develop antidepressant compounds. So it's really not surprise when you look to this whole panorama that our current pharmacology, our current antidepressants, especially the monoamine-based antidepressants, which are the mostly used, have low response rates, and they also have a delay onset. So we need more, we need to understand the biological basis of this condition, and we need different targets. We need to take advantage of this heterogeneity to find different targets and new targets to develop new antidepressant medications. So having this in mind, I'm going to show you just a little bit about the neurobiology of depression. Depression is a highly heterogeneous condition, as I show to you, and it's a multifactorial condition. It has the involvement of genetic factors, epigenetic factors, environmental factors. We have sex differences and many other variables involved in the etiology and progression of this condition. Among those alterations, there are alterations that are already evaluated in neurotransmission, in neuroplasticity, and connectivity in different brain areas. However, nowadays, depression is seen as a whole body disease, and we have alterations in different systems, not only in the central nervous system. And we have alterations in dysfunctions in the stress response system, including the HPA axis, in the autonomic system, metabolic alterations, and of course, we have an involvement of the immune system, which is the focus of my talk today. And the involvement and the participation of the immune system or of immune dysfunction associated with depression is not a new knowledge. So we know that for a really long time, since the 80s, and the studies that actually try to correlate these two things started with observations that depression is more prevalent in patients with chronic inflammatory conditions like obesity, heart disease, and others. Besides that, it was also observed that patients undergoing cytokine therapy for certain types of cancer and for viral diseases actually had depressive symptoms, develop more frequently depressive symptoms. In addition, we observe an increased level of circulating cytokines, actually a low-grade inflammatory response in a subset of patients with depression. And this is interesting because, again, not all patients have this dysfunction in the immune system or these immune alterations. It's not common to all patients, but to a subset of patients. And finally, neuroinflammation is observed in animal models. It's really more difficult to study neuroinflammation in human subjects, but also microglial changes and changes in microglial morphology were observed in post-mortem brain of suicide victims with major depressive disorder. And also imaging studies have shown microglial activation in patients with depression in different brain areas. So we have this peripheral inflammation, we have potentially this neuroinflammation, at least in a group of patients. And so our focus here is to study how we can take advantage to this knowledge and try to focus on different targets and understand how they can modulate or they control emotional behavior. And we focus here on microglia. Microglial cells Microglial cells are the innate immune cells resident in the brain. So they have different functions. They control, they respond, they control the immune response, the immune response in the brain parenchyma. They also are involved in synaptic pruning. They are involved in the control of neuronal activity, and they have multiple effects, multiple actions in the brain. And they control these actions actually by interaction with the surrounding cells. So the microglial cells express wide array of receptors in their membranes, and these receptors are sensible not only to the products that are secreted by neurons and by other cells, but also to lipids and glycoproteins and other molecules that are expressed in the membrane of these cells. So in order to perform these functions and communicate with the surrounding cells, microglial cells use these different types of receptors that are mostly not well studied, at least in terms of mood regulation and psychiatric conditions. There are some receptors that are most characterized, like this CSF1 receptor that is involved in microglial survival and proliferation. We have here purinergic receptors that are sensible to ATP, and they actually can control the microglial response and synaptic remodeling after neuronal activity, changes in neuronal activity. We have here TREM2 receptors that actually can bind to lipids and glycoproteins in the cell surface, in the surface of cells that are dying. So there's they can signal apoptosis and phagocytosis, these dying cells, and we have many other receptors. And the CD300 receptors are really similar to the TREM2 receptor, which is most well characterized and was already involved with Alzheimer's disease. And the CD300 receptor is a family, comprises a family of receptors. There are transmembrane glycoproteins that bind to lipids, including ceramide, phosphatidylserine, lipoproteins, and others. And this figure represents this family, the CD300 family. And in this family, we have both inhibitory and activating receptors. So we have 7 members here, and the CD300A and CD300F are the inhibitory receptors. They have this long intracellular cytoplasmic tail. And the others are the activating receptors. They have a short cytoplasmic tail, and they have here charged amino acid residues in the transmembrane loop where adaptor proteins can bind and start the signaling pathways. So these receptors are expressed in innate immune cells in the periphery as well as in the central nervous system. And they operate, they control microglial function, especially because they operate as inhibitory and activating receptors. So the inhibitory receptors here, including the CD300A and CD300F, as I mentioned, have this long cytoplasmic tail with several inhibitory motifs based on tyrosine, and the phosphorylation of these motifs can recruit phosphatases, and these phosphatases can actually reduce cytokine secretion and reduce the activation of the cells. On the other hand, we have these activating receptors. They are a little bit different, as I mentioned, so they do not have the cytoplasmic tail. Instead, they have this charged amino acid residue that is able to bind adaptor proteins. And here we can see DAP12 and DAP10 that are the adaptor proteins that can bind to these receptors. And this and these adaptor proteins, once they are phosphorylated, they can recruit protein kinases, and they can activate the cytokine secretion. They can activate different cellular response within microglial cells. So this structure is important because it determines the intracellular loop here, and this structure determines the functional properties of this immune receptor, so inhibitory versus activating receptors. And we are interested in the CD300F receptor, which is this one. And the CD300F receptors are classically inhibitory receptors, so they have this intracellular cytosolic tyrosine-based inhibitory motifs. They can recruit phosphatases, but they actually have a dual effect, and this was already demonstrated that they are also able to recruit kinases, including the PI3K kinase here. So they can have some stimulatory effects on microglial cells. These receptors are expressed in central nervous system myeloid cells, especially in microglia and also in perivascular macrophages. They respond to lipids. They can bind extracellular ceramides. They can bind lipoproteins. They can bind phosphatidylserine and others. And they actually, as I told you, are a little bit different from the others because they can display this dual inhibitory or activating activity depending on the context. And these receptors were already evaluated in some pathological conditions. So a few studies evaluated CD300F receptors in pathological conditions, and especially the absence of these receptors potentiates inflammatory and autoimmune disease models. So, and the first studies associating these receptors with changes with modulating in central nervous system responses started a while ago, but we have only a few studies. So the first one was this: the CD300F receptors were neuroprotective and reduced inflammation in an animal model of multiple sclerosis. So the absence resulted in a significant increase in nitric oxide, pro-inflammatory cytokine production, and increase in demyelination in this animal model of multiple sclerosis. And our story, actually not the story of the neuroprotective role of CD300F receptors for us started with this work of our collaborator, Dr. Hugo Pelufo, in 2012. And he showed that the overexpression of CD300F receptor is capable of inducing neuroprotective effects against cytotoxicity and cellular death induced by NMDA administration. So here show that after any NMDA administration, the animals that overexpressed human CD300F receptor had a reduction in the lesioned area in the striatum and also in the cortex. So this pointed to a neuroprotective potential of this receptor. And after that, and more recently, the CD300F receptor was also associated with neuroprotection, and it was associated. It was showed that animals, an animal model of a tau pathology, the tau22 mice, have a downregulation of the CD300F receptors. So this is a more recent study. But despite all this potential and despite all this evidence, no one looked to the possible role of this receptor in psychiatric disorders. So we asked that question. So these receptors are expressed in microglial cells. They are able to control neuroinflammation, but they also have other properties. and maybe they could be interesting targets to mood regulation and to psychiatric disorders. And we started this work. We divided this work in two parts. The first one, evidence from an animal model for transgenic mice, and also the second part, evidence for human studies. So we started this work evaluating the possible alterations in the behavioral phenotype of CD300F knockout mice. So we used female mice, 3 to 4 weeks old, and we tested this mice in these really simple behavior tests. This was the tail suspension test, the forced swimming test. These tests are actually behavior despair tests, and they evaluated the ability of the animals to escape in these acute stressful situations. And here we can see that knockout mice, female knockout mice for the CD300F receptors had an increase in the mobility time in both tests. So this lack of escape-oriented behaviors can be associated with a depressive-like phenotype. And also we have here the sucrose splash test. So we can see that the CD300F mice had a decrease in the total amount of time grooming after the sucrose splash test. Now, after a high viscosity sucrose solution was spread in the coat of the animals. And this is an indicative of lack of motivation, lack of the grooming, the lack of motivation and self-care, and also anhedonia. So these tests suggested to us that female mice that do not express the CD300F receptor have behavior alterations, especially in emotional behavior, that are relevant to mood disorders. And interesting, this behavior is persistent. So we also tested older animals, older female animals with 18 months old, and this phenotype is persistent. On the other hand, when we tested male CD300F knockout mice, we did not find any alteration in these behaviors. So the male mice have other behavioral alterations, especially in anxiety, but not these ones, not in depressive-like or motivation or anhedonic behavior. So this was interesting. We're not expecting this. And together we suggest that the absence of the CD300F receptor in female mice is associated with a depressive-like phenotype. After that, interestingly, the CD300F receptor is among the highest expressed receptor in microglial cells after intraperitoneal LPS administration. And with this information, we performed this other experiment when we treated animals with one dose of LPS, and we evaluated the behavior 24 hours later. And here we can see that in the wild type animals, the LPS induces an anhedonic-like behavior in the splash test, in the sucrose splash test. But this behavior is even potentiated in CD300F knockout mice. So the absence of these receptors potentiate the effects of LPS. And this was also performed in female mice. So with this in mind, and with the idea that these receptors are mainly expressed in microglial cells in the central nervous system, we performed an isolation of these microglial cells and RNA-seq analysis, and from the hippocampal microglial cells from CD300F and knockout mice with and without LPS treatment. And as you can see here, the absence of CD300F was not associated with a huge amount of differentially expressed genes. So when we compare wild type and CD300F knockout mice, and here we can see some panel of these differently expressed genes. There are not so many genes, and most of these genes are not related to a classic inflammatory response. So we have different expressed genes that are not inflammatory cytokines, or the genes that we're expecting to have with the absence of the CD300F receptors. Then when you compare the wild type and CD300F knockout mice after LPS injection, we can see that LPS induces the different expression of a high amount of genes, almost 6,000 different genes. But in the CD300F knockout, only 197 are differently expressed compared to the wild type LPS mice. So this means that, and again, with a few exceptions, no classic inflammatory genes were observed here. So this data suggests to us that the absence of CD300F receptor in these female mice did not predispose the animals toward a pro-inflammatory phenotype. But actually changes a little bit the response induced by LPS. So instead of having a more pro-inflammatory phenotype, we have a different gene expression after the LPS injection. And actually, this next graph that I'm going to show here was an experiment that was performed before. So here we used total extracts for brain and spinal cord, and we found that an increase in the expression of two specific macrophage markers, the CG163 and cyclic 1, with no changes in the other perivascular macrophage marker, CG206. On the other hand, this was just differences in the expression when performed the flow cytometry analysis. There are no differences in the number of positive cells for these markers. So showing that it's not a difference in the number of cells, but in the expression of these markers. So this is a suggestion that we maybe have to look to other cell types that also express CD300F receptors that may be possibly involved in these behavior responses as well. And in addition, we also evaluated the number of microglial cells. So here we can see in adult brain, here in the cortex, we have immunohistochemistry analysis of IBA1 showing an increase in the number of microglial cells in CD300F knockout mice. And also by flow cytometry, we show here an increase in the number of microglial cells in the hippocampus. It's important to mention that this is not related to apoptosis in wild type mice samples. And also in microglial cell cultures from adult mice, we also observed this increase in the number of microglial cells. And this increase in the number of microglial cells is something that probably the animals acquire with time, because when we perform the evaluation in P12 animals, in the cortexes of P12 animals, which is where microglia already stopped the proliferation, we did not observe these changes between wild type and knockout mice. And this is interesting because, and when you look to some studies with microglial in patients with major depressive disorders, we actually can see an increase in the activity or an increase in the number of these cells. So it corroborates a little bit the literature so far. And So and one of the genes that were differently expressed, and it was really interesting for us after the RNA-seq analysis in hippocampal microglial cells after LPS injection, were genes associated with metabolism. So we have here some of them. So we found a downregulation in the CD300F knockout mice hippocampal microglial after LPS injection. We had a downregulation of different genes involved in glucose transporter, in glucolysis like exokinase 1. We have genes, the downregulation of genes involved in beta oxidation. We have genes, the downregulation of genes involved in Krebs cycle and also pyruvate transporters. So it looks that after LPS injection, the metabolism, the genes involved in basic metabolic pathways in microglial cells were downregulated. With this result in mind, we actually performed some functional experiments here. So we used these isolated microglial cells in the seahorse experiment to evaluate the metabolism. And as you can see here, the knockout mice after LPS injection, the microglial cells, hippocampal microglial cells from knockout mice had a reduction in the basal oxygen consumption rate, and also in the glycolysis here, evaluated by the amount of extracellular exit. And here, it's important that when compared to the wild type mice also exposed to LPS. Moreover, the glycolytic shift that is normally observed in wild type mice, and here when we use a mitochondrial stressor, FCCP and oligomycin, we have this shift towards an anaerobic metabolism that is common. In knockout mice exposed to LPS, these microglial cells lose this ability to shift towards an anaerobic glycolysis. So this result was not associated with changes in the number, in the mass of the mitochondria, but it was highly suggestive to us that the absence of CD300F receptors actually has a major impact on microglial metabolism. And this can be one of the possible mechanisms involved in the other characteristics that culminate with changes in emotional behavior observed in these female mice. And so this is the preclinical part. So just to summarize, female CD300F knockout mice had a depressive-like phenotype and an anhedonic phenotype. We did not observe, with really few exceptions, we did not observe any changes in classical inflammatory markers, but we do observe an increase in the expression of genes associated with perivascular macrophages, and also we do observe changes in microglial number, an increase in microglial number. And when we look to the microglial cells of these knockout mice, we again do not observe major changes in inflammatory genes, but we observed the downregulation of several genes involved in key metabolic pathways, including glycolysis, beta oxidation, Krebs cycle, and we do observe a fail to adapt. So the absence of a shift towards a metabolic shifts after a mitochondrial stressor. So we also conducted a parallel study, a human study, and I'm just going to show briefly our results. We have the CD300F receptors is located on the chromosome 17, And there are a few, I think maybe 3 or 4 studies showing that this lossy where the gene is located was already associated with some inflammatory conditions, including general inflammation, but also colitis and multiple sclerosis. But there are not enough studies evaluating polymorphisms or changes or mutations in these genes in association with diseases. And so we evaluated this single nucleotide polymorphism. There is a non-synonymous substitution. We have the exchange an arginine to a glutamine in the cytoplasmic tail of the receptor. So, and this single nucleotide polymorphism is relatively frequent in the population. We have here a frequency of 12% in the European population and 29% in the American population. So we evaluated the frequency of this single nucleotide polymorphism in a cross-sectional population-based study that was conducted in the south of Brazil. And here, we had in our sample 625 controls without major depressive disorder and 485 patients with the diagnosis of major depressive disorder. So as you can see here in this table, the frequency of the T allele is it's small in the population. So we used here the dominant model. So we grouped the individuals carrying the T allele, both the CT and the TT individuals. And as you can see here, the presence of at least one copies of the T allele was associated with a reduction in the prevalence of major depressive disorder in this population. And here we can look directly to this column. We have the adjusted analysis showing that even after adjusting for other factors like ethnicity, age, and other demographic variables, the presence of at least one T allele is associated with protection against major depressive disorder, with a decreased risk against major depressive disorder. One thing that we did next was to stratify this population. And here we have results in women and in men. And as you can see in the last column here, this protection remains significant in women even after adjustment. So the presence of at least one T allele was associated with a reduced risk for depression. But when you look to the male population, this effect was lost. And you have some limitations of this analysis. For instance, our population has more women than men, especially in the major depressive group. So we lose a little bit of the power of analysis when we conduct this stratification. But I think that's a good evidence that corroborates our previous preclinical data, showing that maybe there are sex differences in the way CD300F receptors are associated with behavioral modulation. So we also evaluated peripheral levels of inflammatory cytokines in these patients, and we did not observe any association between inflammation with any of the genotypes of this single nucleotide polymorphism. So finally, and this work was conducted in Barcelona by one of our collaborators, and they asked the question, what's the functional importance of this single nucleotide polymorphism? So they actually used COS7 cells that were transfected with the human CD300F gene. And here, the cells were stimulated with different compounds that actually activated protein kinase A, protein kinase C, and phosphatases. And here you can see that the PMA, which is a PKC activator, actually increases the phosphorylation, the serine and threonine phosphorylation in the cytosolic loop of this receptor. And on the other hand, when they transfected the cells, so here is the transfection with the human receptor, and showing that the phosphorylation after PMA induction. And here, when the cells are transfected with the human receptor containing the single nucleotide polymorphism that we evaluated, this phosphorylation is lost, suggesting that this exchange, this amino acid exchange, is in the intracellular tail of this receptor in a region that is modulated, phosphorylated through a mechanism dependent on PKC activity. So we don't know so far the other implications of these signaling alterations, but you can say that this polymorphism that we show that confer protection against major depressive disorder in women is also has a functional effect. So it's in a PKC site of phosphorylation of this receptor. And so just to summarize all the results, we have this preclinical evidence using CD300F knockout mice, and we show that these receptors have behavioral alterations especially in emotional behavior, and here depressive-like or lack of escape-oriented behaviors in the forced swimming test and tail suspension tests, and also anhedonic behavior, and this was observed only in female mice. When you look to total brain and spinal cord extracts, we have some discrete changes in some inflammatory cytokines, which I did not point it. So a discrete increase in interleukin C in interleukin-1 receptor antagonists, a decrease in interleukin 10. We also observed a decrease in the levels of noradrenaline in the hippocampus, which I did not show you today. But not major inflammatory activation. When you look to the changes in microglial cells, we observed an increase in the number of microglial cells in these knockout mice. We also observed a change, a downregulation of important genes associated with microglial metabolism, and this absence in this lack of capacity to change after a mitochondrial stressor to an anaerobic metabolism. So this was our preclinical findings. And in humans, we observed that this single nucleotide polymorphism in the CD300F receptors, which is an amino acid exchange in the cytoplasmic tail of the receptor, confers protection against depression in women. And this site is possibly a site where protein kinase C has an impact and is capable of phosphorylating this receptor. So we don't know the consequences of this yet. These results, most of these results were published last year. So here I want to thank all the collaborations. It was a huge collaborative effort. So if you guys want to check. And also we have some perspectives now. We are currently in my lab evaluating the role of these receptors in neuroplasticity and neurogenesis. So we are trying to see if one of the mechanisms is actually the modulation of the synaptic remodeling and neuroplasticity in the hippocampus. And the other perspective that is being conducted in Dr. Hugo Pelufo's lab in the Pasteur Institute, and they are generating these different types of conditional knockout mice. And one of those is actually humanized transgenic mice expressing the the human single nucleotide polymorphism in the CD300F receptor. So we are pretty excited to see how these mice behave in these emotional related tasks. And here, I think that the most important slide, I would like to thank a lot of people, especially the group here in the Pasteur Institute from Montevideo that is coordinated by Dr. Hugo Pelufo and also Dr. Natalia Lago. So as I told you in the beginning, they are actually the immunologists involved in this research, and they are actually the people who with a long trajectory studying this CD300F receptor. So we are more focused on the psychiatry disorders and the modulation of emotional behavior. So we think most of these experiments that I showed today were conducted there. I also want to thank the group from the Catholic University of Pelotas in Brazil, especially Dr. Gabriele Ghisleni. They actually coordinated the human study, so they are really good in epidemiology and clinical trials and human studies with psychiatry disorders. So it was really good to see our preclinical evidence being translated in human evidence. And also my collaborators in the Federal University of Santa Catarina, especially Dr. Ana Lúcia Rodrigues and Dr. Patricia Bocardo. Patricia is helping us with this neuroplasticity and neurogenesis evaluation now. And here's a picture. Now, I also want to thank Dr. Fernanda Kaufmann. So most of this work was part of Fernanda's PhD thesis. And here is a picture of me, Fernanda, and Hugo, which was Fernanda's co-supervisor during her PhD presentation. And we are very happy to be awarded an honorable mention in the CAPS Thesis Award in Brazil last year. So Fernanda is now a professor at Federal University of Pelotas. And here my research group in one of our virtual meetings this year. So I want to thank everyone for the efforts and their dedication during these really, really difficult times. And finally, for the financial support. And again, I want to mention the importance of the International Brain Research Organization that allow us to collaborate and to interact with a lab that is really close to us. In Uruguay and Brazil, we were really close, but IBRO gave us the opportunity to collaborate and to look at these questions from different perspectives, from an immune-related perspective and from a neuroscience perspective. So I think that's really important. So thank you very much. And I'm open for questions.
Thank you so much for a wonderful talk. I'd like to remind all attendees that there is a Q&A section where you can put in questions, and we will go to the questions afterwards if Dr. Kaster can take them. I have some questions, but I think let's start with the ones in the Q&A. And since not everyone can speak, I'm going to just read out these questions. The first question comes from Nanu Pano, I hope I pronounced that right, who says, how can we evaluate the expression of CD300F where genetics tests are not always available? What will be an alternative way to regulate mood in patients with major depressive disorders?
Okay, let me see if I got the first time. How to evaluate the expression of CD300F receptors?
Question is more that if you cannot evaluate the expression of 300F because it's a place where there are not genetic tests, where genetic tests are not easily available, would there be an alternative way to address major depressive disorder? So it's a pretty broad question.
Yes, this is actually complicated. First, the antibodies for the evaluation in peripheral cells, we still do not have these results. We are also interested in see if we have peripheral changes in the expression of CD300F receptors in peripheral immune cells. and maybe if this can be an indicative of something going on in the central nervous system. But for now, the antibodies are not very good, and for the expression, we do not have this result so far. And the other thing is that we do not understand yet how these receptors can actually modulate behavior, and probably the effects on different cell types are different. The effects are different. So we have here some evidence pointing to microglial cells, but also our evidence opened the idea that maybe perivascular macrophages can also be involved in some of these effects that we observed in the knockout mice. So I think that's, I don't have a satisfactory answer for you, but I think that's just the beginning. So I think that depression is actually a multifactorial disease, and there are a really, really high number of genes with a small effect that are they are involved in depression. So I cannot say that only this mutation in the CD300F receptors can be associated with the disease. We have to replicate this study in different populations to see if the effect is still there. And we also have to look to a broader array of genes and of markers. So I think that's maybe in the future, hopefully in the future, we can contribute more. But for now, our idea here is just to start digging different targets and starting to see how these different targets can be involved in this disease.
Thank you so much. So a second question from Alexis Danino is, are CD300F knockout mice vulnerable or resilient to stress? And as a follow-up to that, is there any alteration in the social behavior of CD300F knockout mice compared to wild type?
That's a very, very good question. Actually, we are doing that. We plan to do that. So we plan to use the chronic social defeat stress to see in a more robust model how this animal responds. So we use LPS a little bit with this idea that is we know there is LPS. It's not the best model to study depressive-like behaviors, but we use the LPS stimulation a little bit with this idea. But now we are going to use more robust models like the chronic social defeat stress. The thing is that we already have the male chronic social defeat stress going on in the lab. So we have to test the CD300F knockout mice, but the male model is already okay. We are now working on the development of the female model, which was a little bit tricky since female mice are not so prone to attack as male mice. So, but this is the model that we are going to evaluate to see if we have resilience and susceptibility. And yes, I think that's the next steps. And we expected to see the similar thing that we saw with the LPS. So the knockout mice are more prone to vulnerability than the wild type mice. But let's see.
Thank you. We have another question. Actually, it's three questions from Sabrina Lisboa, who starts with saying, wonderful work, thank you, about the expression of pro-inflammatory gene expression in the RNA-seq analysis and the metabolic changes. I have three questions. Do you believe that CD300F is involved in the priming state of microglial cells to subsequent stimulus? Do you have evidence of which intracellular signaling modulated by CD300F is linked to this metabolic pathway? And is this metabolic pathway alteration directly involved in a primed state of microglia? So yes, I can go back to those three questions later if you like, but really about the priming of microglia and the response.
Yes. Also, they're really, really good questions. we believe that the changes, some of the gene expression that we won't perform the RNA-seq analysis in the after LPS administration. So we do believe that most of these changes occurs after a stimuli or after some type of priming. We don't believe that we have many basal changes in the metabolic microglial metabolism in wild type versus CD300F knockout without any stimuli. And I was talking yesterday with Hugo about that, and he told me that they are experimenting in bone marrow macrophages because of the number of the cells, and they are not expecting to see any basal changes without the LPS administration. So in the RNA-seq analysis, we did not observe major changes in these metabolic pathways without the LPS stimuli. So I think that the comprehension of how the microglial metabolism is affected is something that we are still looking, but probably after this occurs after a stimuli, and it's not so much in basal conditions. And I don't know if I get all the three, probably not.
I think you did touch on all of them without, she did ask about the signaling pathways, and you mentioned that there's not much change.
Not much, but another signaling pathway that I did not mention that we're actually Dr. Pelofur is going to pursue in the future is the changes in autophagy. So the RNA-seq data found this downregulation in the metabolic pathways, but also dramatic changes in autophagy in the microglial cells. So we are probably are going to pursue these two paths in the near future, hopefully.
Wonderful, thank you. Since while we wait, if there's somebody else who needs to ask a question, maybe I can take this opportunity to ask you a couple of questions. One actually leads off of what you've just been talking about this necessity for LPS to actually really highlight the differences between these two genetic states and really sort of emphasize the predisposition towards mood disorders. Do you think this has implications for human patients in terms of susceptibility to infection and the appearance of a mood disorder post-insult, post-inflammatory insult in some way?
Yes, I think that this is it. I think that of course inflammation is a good and a positive response, and we are now experiencing after the COVID-19 or during COVID-19, we have some initial results showing that actually the infection itself can be a trigger from some changes in behavior and for the initiation of mental illness in some people, but this is really new results. But we know for a really long time that despite the fact that the inflammation is a good and a positive response, it is a stressful thing, and how our body and how cells deal with, especially with the resolution of the inflammatory mechanisms, is important, because chronic inflammation is associated with actually changes in neuronal circuits and changes in neurotransmitters. And we have this connection between chronic low-grade inflammation or microglial reactivity and changes in brain circuits modulating behavior. So I think that this is the question. We are all going to experience stress. We are all going to experience infection. This is something that we cannot avoid. So if you understand how some of our cells or our body can deal with that, and how these resolution mechanisms are in place, how they are different in some individuals, maybe we can, at least for a subgroup of patients, not all depressive patients, not all patients with major depressive disorder are equal. There are patients that have an inflammatory compound and others that do not have. So at least for some groups of patients, we can try to target therapies that are going to be more effective than just a general therapy.
Wonderful, thank you. Maybe I could again ask another one that is really fascinating. Your results, both in the human and the mouse, about the susceptibility of females in the case of the humans lacking the T allele versus as well as in the knockout mouse model. Absolutely fascinating that the females are so much more susceptible than the males. It seems like a really robust result here. So I was wondering whether you have any hypotheses about why this is that the females are specifically susceptible here.
Yes, thank you very much. This is the question of the day, of course, for us too. And I'm sorry to say that things that are still going on. We have planned experiments because last year the lab was mostly closed. So we were impressed with these dramatic sex differences. When you look to the human data, I want to be very, very careful because it does need replication. It does need a higher power of analysis. So it does need a lot of things to actually say that. But for the animal phenotype, it was really clear. So what we know so far is that depression is a different disease in men and women. So the results from gene expression in animal models, in stress models, and also in human individuals shows the completely different patterns of gene expression in men and women with major depressive disorders. So maybe this is one of the clues. And actually there is one human study showing that in men, there are higher expression of genes associated with microglial function in men with major depressive disorder, but not in women. And many of these responses go in opposite directions. So I do not have the answer for that. But I think that's we have to start treating this as completely different disorders, not only from the clinical point of view, because men and women also have maybe the prevalence of different symptoms and different clinical manifestations, different comorbidities, but also different biological mechanisms involved in this condition. And we have to investigate that.
Thank you so much. Yeah, this is really going to be a fascinating set of studies and results coming from your work. There's one more. We're almost out of time, so I'll just try and catch the last couple of questions. This is again a question from Sabrina Lisboa, who asked, this receptor is mostly expressed in microglial cells, but when using knockout mice, the receptors are deleted in all cells, including during neurodevelopment. Do you have evidence from conditional knockout mice to delete CD300F only in microglial cells to see if it's actually microglial mediated, and also if it is not something related to the absence of this receptor during brain development?
That's also a great question. The animals are being prepared. So we are, Dr. Hugo Peluf is actually generating these animals. And of course, it was a huge concern. One thing that I can say is that the global knockout mice do not have any huge physiological, inflammatory, or alterations in basal conditions. So we did not observe changes in inflammation or changes in the number of immune cells or any physiological changes. But of course, to actually answer that, if it's microglial cells, and now even if it's the role of the perivascular macrophages, which is something that's really interesting, and it's really new in this field, the contribution of this other innate immune cells in the central nervous system that are not microglial cells. And this is another thing that we are going, hopefully we're going to ask soon.
Wonderful, thank you. We have a couple of questions in the chat, which I believe you've already answered. What would be the clinical significance of this model, and what is the rationale for gender specification during the study? I think we've been discussing that for a while, but if you'd like to add more, we can pause, but I believe you have already addressed these topics.
No, just regarding the clinical signification, I think that we need to be very, very, very careful, because as I told you, it's just a small piece of evidence, and depression, the involvement of multiple genes are associated with the major depressive disorders, and the contribution of these genes are different, and we have to be careful with the evidence and actually understand what the evidence is telling us, which is maybe it is a good target, but we have to dig deeper to see the exact contribution.
Thank you. Those were questions from Rimpi Aurora. I didn't mention her name, I apologize. And for a final question, I know we are over time, but maybe we can take one final question from Carol, who asks, could the data on changes on metabolic fitness relate to other tissue macrophages, including the inflammation response in those tissues?
The data from the metabolic pathways were derived from RNA-seq from microglial cells, isolated hippocampal microglial cells. So let me think. Maybe if there is an indirect contribution, we cannot say for sure, but maybe to look at this, I don't know exactly how you should look at this, but yes, I think we cannot discard an indirect contribution. So after we have the global knockout mice, we have the 24 hours of LPS, so which can generate different signals and different effects on these microglial cells. And we cannot exclude that there are other players in these changes in metabolism, of course, and probably they are.
Thank you so much, Dr. Kaster. We're out of time now. It's 11 minutes past the hour, but I just wanted to take this opportunity to thank you again for a really informative talk, a wonderful talk. There have been several messages in the chat thanking you for the talk and saying how interesting it was and how much they enjoyed it. So please join me in thanking virtually our wonderful NextInNeuro speaker, Dr. Manuella Kaster.
Thank you very much for the opportunity. It was really great, and I'm available if anyone has more questions. Feel free to contact me. And thank you very much for IBRO for everything.